Hello my dear students and welcome back to Excellence Batch, and I am your Diksha ma'am. So here we have started the chapter Evolution. In the previous class we have studied that how the universe origin took place around 20 billion years ago, how different planets were formed and what were the different theories given by various number of scientists on origin of life. So today we are going to talk about something which is little maybe with the proofs, right? So we were saying yes, evolution takes place, this is what evolution is, this and that, but what actually and how actually and where actually the evolution is taking place? So let's give some proofs, right? So today we are going to talk about the evidences of evolution, where we can see yes, actually the evolution is taking place. So we are going to talk about various kinds of evolution's evidence, right? But today we will be talking about only two evidences. The first is anatomical evidence, second evidence is biogeographical, third is embryological, fourth is fossil, and fifth is biochemical. So we are just going to talk about these two today. But these all evidences give us a proof that yes, evolution is taking place. So let's get started with the anatomical evidences. Anatomical evidences, they are also known as morphological evidences. That means here we are going to talk about the body structures, the anatomy, the morphology, that how the organism looks externally as well as internally, or how the body of the organisms, they tell us about the evolution. So let's see how. So first thing that we are going to discuss is homologous organs. What are homologous organs? These are certain organs in which the structure is altogether similar, but the function they perform is different. For example, you have man, cheetah, whale and bat. The common thing in all these are they all are vertebrates. What are they? They all are vertebrates. If you'll see closely, their forelimbs are these limbs. The forelimbs, they match, right? Somehow they match, they are similar, but all are performing different function. How? Man is using the forelimb to hold the things. Why do we use our hands? We use hands to hold objects, right? The cheetah's forelimbs are adapted for running. The whale's forelimb are flippers, they are adapted for swimming. And bat's forelimbs have been modified into wing-like structure that help them to fly. So basically homologous organs are all those organs where structures are similar, but the function these structures they're performing, that's different. Right? So the first example is forelimb of vertebrates. Like in man, the use of forelimb is to grasp the object. In cheetah, the forelimbs are adapted for running. In whale, it is adapted for swimming. All are vertebrates. If you see, all are vertebrates. In fact, all are mammals, right? And the last one, bat. In bat, they are adapted for flying. So let's read the NCERT line. The same structure developed along different directions due to adaptations to different needs. This is divergent evolution and these structures are homologous. So now how did these structures they evolve? As you know, they all are what? They all are mammals. Right? So technically if you'll see, the origin of all of these is same, they all are vertebrates. But because they have went to various different habitats, according to their habitats, they modified their forelimb. For example, cheetah had to run, so cheetah adapted their forelimbs in a way. Whale was in water, so whale adapted their forelimbs in different way. Bat needs to fly, so bat has adapted the forelimb in different way. So the origin of all were same, they all were mammals, but they have adapted, they have diverged themselves, they have shown an evolution. In that evolution, what's evolution? It's a change. They have shown a change in which they have adapted according to their habitats and their surrounding. So such kind of evolution is known as divergent evolution. What do you call this evolution as? Divergent. That they're diverging from the common origin and going to the places. And in that places, they're showing the changes, their evolution. Fine. So always these structures, these are homologous organs, and what kind of evolution they are showing? They are showing divergent evolution. Let's see their example in plants. In plants we have bougainvillea. The bougainvillea's thorn and cucurbita. Cucurbita is a family of cucumber, right? If I'm not wrong, because I'm not a botanist. Cucurbita, they have tendrils. If you'll see, thorn in the bougainvillea is performing a function that it will protect the plant, right? And tendril has a function that it helps them to climb, right? So both are performing different function, but they both are axillary in position. That means their position and origin is same. Right, their structure is same, but the function they're performing is different. So that's an example in the plants. But always homologous organ will show the divergent evolution. Their origin, their structure was same, but these organisms, their structure was modified in a way, they're performing different function. Okay, all right, so that's the first example in anatomical homologous organ. Let's solve the question. One trick to solve the analogous and homologous organ, another we'll be discussing is analogous organ. Many of you get confused in homologous and analogous organs questions. You don't know how to solve them. So one trick that I can tell you is that whenever you have the answer or question related to homologous, all the organisms should belongs to the same phyla or group. Their origin should be same. Like in this one, you can see they all were vertebrates, so they belong to the same group. Okay, let's solve this question. Among the following set of example for divergent evolution, select the incorrect option. Forelimb of man, bat and cheetah. Heart of bat, man and cheetah, eye of octopus, bat and man, and brain of bat, man and cheetah. So straight away, even if you don't know the all examples of homologous organ, you can just see that octopus is a mollusk, bat and man, they are mammals. So their origin is different. And when origin is different, that means it's not homologous. It's not showing the divergent evolution. So answer is C, this is incorrect. Now what about the other example? Brain of Batman and cheetah, heart of Batman and cheetah. They are the other examples of homologous organ. So in homologous organ, the heart and brain of vertebrates, they also show divergent evolution. How? Let me tell you. For example, if you'll see the development pattern of the Batman and cheetah, you will see all these organisms, they have around similar pattern of a brain, similar structure of a brain, but the function they're performing is different. R is more for logic and reasoning. Like cheetah's brain is more for preying, catching the prey and hunting and all. So the structure is literally the same, but the function they're performing for is quite different. They are not studying, right? They're not studying right now homologous and analogous organ. So these are the example of other examples of homologous organ. Alright, let's move further and talk about analogous organs. Let's talk about analogous organs. Okay, so these organs are organs that are not anatomically similar, but they perform similar function. So here structure is different. Structure and origin, they both are different, but the function they perform is same. But the function they perform is same. So we have a lot of example. First example is wing of a bird, insect and bat. Alright, so what's the common thing in them? The common thing in them is they help in flying. Even if you take a wing of a bird, they are flying. Take a wing of an insect, they are flying. Take a wing of a bird, they are also flying. So the common is the flying. But they are coming out from the different origins. So that means they are converging here. Converging means to come up and get here, right? So they are converging here at a common function. So such kind of evolution these structures show is convergent evolution, which is opposite to the divergent evolution. In divergent evolution, what have we done? In divergent evolution, the organism's origin was same, but they diverged. They went out somewhere, right? And they adapted themselves according to the surroundings. In convergent evolution, their origin is different. They're coming from different origin. Maybe that's the origin of someone else. That's the origin of someone else. Maybe that's the bird, that's the insect, and that's the mammal. So here it is coming and they are performing the similar function. That's a convergent evolution. So this is the divergent evolution and this is the convergent evolution. Okay, all right. Moving further to the example, some more. Stem of a potato and root of the sweet potato. Both are storing the food. Both function is to store the food. But this is the modified stem and this is the modified root. Flipper of dolphin and penguin. Penguin is a bird and dolphin is a mammal. So the groups are different. So origin is different. But both have flippers that help them to swim. Eye of octopus and mammal. We both have eye and the structure of a retina is quite similar and it helps in vision. So that's why the eye of octopus and mammals, their function is same but the origin is different. Octopus is a mollusk and mammal are mammal. Fin of a shark and whale. Both look like similar, right? Little similar. But shark is a fish. It's a cartilaginous fish. Both fin, they help in the swimming. But whale is a mammal. Whale is a mammal. So you can see here their origin are quite different. The groups are different, but the function they are performing is quite similar, right? Okay. All right. So let's talk about analogous structures are a result of, let's solve this question, stabilizing selection, divergent evolution, convergent evolution, shared ancestry. So we are talking about analogous organs. Analogous organs never share their ancestry because shared ancestry means their ancestors are same. Aves and insects, they are quite different from the ancestry. So they have different origin. They cannot be same. So this will not be the answer, although you know the answer because we have just done the convergent evolution. Divergent is shown by homologous and stabilizing selection is a kind of natural selection which you will be doing in the Darwin's theory. So answer to this question will be C, the convergent evolution. Okay, next. Which one of the following options gives one correct example each of the convergent evolution and divergent? So you just have to find the correct one. Convergent evolution says that bone of forelimb of vertebrate is example of convergent. Wings of butterfly and birds, thorns of bougainvillea and tendrils of cucurbita, eye of octopus and mammals, eye of octopus and mammal, bone of forelimbs of vertebrates, thorn of bougainvillea, tendril of cucurbita, wings of butterfly and birds. Simple. We have done in convergent evolution, eye of octopus and mammal both belongs to different group. In divergent, forelimbs. So answer is straight away C. Others are wrong. You just need to know the examples, or somehow if you don't know the example, you need to know the basics first. Okay. All right. Next. Forelimb of cat, lizard used in walking, forelimb of whale is used in swimming, and forelimb of bat used in flying are an example of. See, the cat, lizard, they are walking with their forelimbs. The whale is swimming and bat is flying. So all belongs to one group, and that's vertebrates. So what is this? What they are showing? They're not talking about the evolution. Have they mentioned that they're asking you about the evolution? No, they're not asking about evolution. They're simply asking about the structure. So answer is C. They all are showing homologous organs. The function is different. The statement is simply saying the function is different. How? One is walking, another is swimming, another is flying. So the function is different. Straight away everything is done here. But they're not asking you about the evolution. They're asking you about the structure. So answer is homologous organs. Okay. Alright. Next. The eye of octopus and the eye of cat show different patterns of structure, yet they perform similar function. Very true. If you have an eye of a cat, which is a mammal, and eye of the octopus, they have a kind of a similar structure, a different pattern of structure, but the function is they're similar. So that means that is an example of, if you straight away learn it, then it's an example of analogous. And analogous shows which evolution? Convergent. Let's find it out. Not the homologous. It's the analogous. And with analogous, which evolution occurs? Convergent. So answer to this question will be B. Okay, simple. Next. Which of the following structures is homologous to the wing of a bird? Okay, flipper of a whale, wing of a moth, dorsal fin of a shark, hind limb of rabbit. First of all, they're asking you about homologous. So that means their origin should be same, that organism should belong to the same group. So rabbit is a mammal, whale is a mammal, moth is an insect. So because this is from different phylum, it will not be the considered case. Then shark is a fish, so it is also not from the similar origin. Alright, now let's talk about rabbit and whale. Now tell me which one will be a homologous organ to a wing of a bird. The wing of a bird is a modified forelimb, not hind limb. Modified forelimb. And flipper of a whale is also modified forelimb. So this will be the best answer. Answer is A. That is hind limb. Hind limb means the one which is after the forelimbs. Okay. Next is in the morphological evidences, vestigial organs. Now what are vestigial organs? Vestigial organs are rudimentary organs or organs which were functional in ancestors, but non-functional in us. So there were a lot of organs, might be they were functional, they were working something, they were for a purpose in our ancestors. But because we have undergone a lot of evolution through these years, million of years, they were not at all useful. So with time, these organs, they get rudimentary or they get non-functional, they get vestigial. So vestigial organs are organs which are not at all functional right now in your body, but might be in your ancestors, they were functional. Let me give you a very good example of our body, vermiform appendix. It's written here, you can see. I can also write it here if it's not clear. Vermiform appendix is, if you have done the digestion and absorption chapter in class 11th, you have seen it's just present with the cecum of large intestine. It is said that the function of vermiform appendix in your ancestors was, it was producing cellulase enzyme. Our ancestors were herbivorous, they didn't cook the food. So to digest the uncooked food, to digest the cellulose of the plants, we needed an enzyme cellulase. So right now we are cooking the food, we eat all the plant-based food cooked. So that get digested with the, because easily, they get digested easily. Why? Because when you cook the food, plant-based food, the cellulose get disintegrated with heating. So we don't need to digest the cellulose nowadays. Okay. Another is the nictitating membrane. Nictitating membrane is the third eyelid It's functional in the birds, it's functional in the some reptiles, amphibians, it's functional in the fishes. So if you'll see your eye, you can find a very small nictitating membrane right here, right? So that's vestigial in us, it was functional in our ancestor. Same canines, pointed canines are no more useful to us. Similarly, these muscles, these six-pack muscles or these muscles around pyramidalis muscles here, segmental muscles on abdomen, these are basically the six-pack muscles. These are the muscles present near obliques, right? Caudal vertebra. What are caudal vertebra? Tail vertebra, coccyx. Our ancestors, they had tail, but we don't have, so it's vestigial in us. Hair on the body, excessive hair were present in chimpanzees, and chimpanzees are considered to be our ancestors, but right now we don't have hair. Can you see any hair on a female's body? Barely, right? A small amount, yeah, they're present, but not in a large quantity, right? Then males have mammary glands which are non-functional. In our ancestors, some male had functional mammary glands. And then the muscles of the ear, these auricular muscles. What are these muscles? Auricular or ear muscles. Ear pinna muscles. Basically, these are the muscles of ear pinna. Our ancestors used to move their ears so that they can collect the sound in the forest. But right now we don't need that, right? So these muscles, they are vestigial. I can bend my ear like this, but by the effort of my hand when a force is applied. But I can't do it myself. So these muscles, they are vestigial. They are no more functional. So somehow these vestigial organs, what they are telling you? That your ancestors were there somehow. In them, these organs were functional, but with evolution they got a change and they are no more functional in you. Or if I say I have tail vertebra, what does it signifies? It signifies might be my ancestors were the one with the tail, right? So this is how it explains a little bit about evolution, and it's more things were clear when we see the cases of atavism. What are atavism? Atavism is reappearance of ancestral characters. For example, I say a lot of organs in our body is vestigial. What if I say all these are vestigial? What if in some people these are functional? So this also tells us that yes, these were functional in our ancestors. If accidentally they got functional now, might be they were present in our ancestors. For example, this man was born and he was born with a lot of hair on his face, right? So excessive hair is one example of atavism. Another, some people can move their ear muscles, these muscles. So movement of ear pinna, that means their muscles are functional. Some people, they get their third molar very early and functional. That's the wisdom tooth. Wisdom tooth is also one example of the vestigial organs, right? I can write it here. Wisdom tooth, third molar, right? Then you must have heard that a baby is born with a tail. So that's also reappearance of ancestral character. So tail in a newborn baby. So this also tells us that if these characters are somehow accidentally are formed in some people, that means somehow they are in our genes, and the genes, we share them with our ancestors. Okay, so that's about the another morphological evidence of evolution, that's atavism. Let's talk about stesigenesis. That's the another anatomical evidence. What is stesigenesis? Stesigenesis is a state where no evolution takes place, or we also call it as static evolution. Static is something that's not moving. Okay, So in some organism, why do we always assume that organism must have done evolution in the due course? Why? There can be chances that some organism, they didn't get any evolution, they didn't get any change. What causes evolution? The change in the habitat, the change in the environment, right? Whenever there is a change in environment, you change. For example, if right now you are living in house freely, nicely, eating house food, and mother is cooking good food and you are eating, and then you have to go to hostels, and in hostels you don't get good food, then you'll be eating food. So this is how you change, and then you will make peace with that food. First of all, you will not eat food nicely because you don't find it good, because for the throughout your life you have eaten your mother's cooked food. But then after some day you will say, no, I have to eat this, I have no other option. But after some days you will start liking that food. So that's the evolution that's occurring. Okay, that's a change that's occurring. I just can't say evolution in terms here, but yeah, that's a change that's occurring. So similarly, you got that change because there was a change in environment, change in the habitat. Fine. What if you don't go to hostel, you just remain in home, and your college is just nearby? Then you will never get that change in your life. Okay, so there are a lot of animals that don't get inappropriate condition, they don't get changing environment, so evolution didn't took place in those organisms as well. So that is what stesiogenesis is. It's a process where no evolution has taken place. So are there certain organism which has not done any evolution? Yes. So we have king crab. We have king crab, Limulus. Limulus is a king crab who has not done any evolution so far in itself, right? Another is Latimeria or coelacanth. Latimeria or coelacanth is a lobe fin fish. You will not find any fish which have lobed fin like this. You will never find any fish. You might find the fish with such kind of fins, but you will never find the fish which lobe fins have, right? So this fish was first seen in 1938 in South Africa waters. And then this fish was not seen. So it is somehow said that if this fish still survives and have lobe fin, so might be there are fishes that were earlier fishes, it is supposed that they had lobe fins. So this fish has not undergone any evolution and it's doing its own work like that. Okay, so yes, there are organisms which have not undergone any evolution so far and they're living happily like that. Fine. So this was about stesiogenesis. Let's talk about the connecting links, the last morphological evidence. So what are connecting links? Connecting links are certain organisms which are a link between two groups or two phylums. For example, if I find one organism that does not completely fits into phylum 1 and does not fits into phylum 2, then what does it tell? It tells that it's an organism which is doing a little change, which is undergoing evolution. So it's an example that when the evolution took place, it took place from phylum 1 to phylum 2. This is how organisms, they originate. That means this organism is still under conversion, and then it will later on has become something that gets into the phylum 2. Fine. For example, let me give you an example. Peripatus. Peripatus is an organism that is a connecting link between Annelida and Arthropoda. So it contains the characters of both the phylums. So it's a kind in a transition. It contains the character of both. So this tells that organism, it moved from Annelida to Arthropoda. And this is how the evolution took place. So whenever you are studying animal kingdom, you always start with the simplest organism. First Porifera, then Coelenterata, Ctenophora, Platyhelminthes, Esculmenthes, then Annelida, then Arthropoda, then Mollusca, then hemichordate, then Echinodermata, hemichordata, and then you go for chordates. So why we are reading this in this sequence? Because it tells about the evolution. Right. Similarly, we have Neoplina. Neoplina is one organism that's again the connecting link between Annelida and Mollusca. We have an organism Platypus. Platypus is egg-laying mammal. So it tells that somehow we have a common ancestry with reptiles because we are laying eggs and we are mammals. So reptiles were laying eggs and mammals, they don't lay eggs. So it's in between them. So platypus is egg laying mammal. Right. Then we have hemichordata. Hemichordata is a connecting link between non-chordates and chordates. Non-chordates and chordates. Fine. Then we have one more example, that is Archaeopteryx. Archaeopteryx is basically not the living organism. All these are living organism. This organism's fossils were found, and it was found out that it's a connecting link between reptiles and aves. So this one is not living, only fossil was found. So any connecting link which is not living, of which we only have fossils, we call it as missing link. So Archaeopteryx is a missing link. It has characters of both reptiles and aves. How? Like reptiles, it has like teeth. Like aves, it has wings. It was able to fly, right? So this is how it has a lot of other characters of reptiles and aves. And this tells us about that this organism is kind of a transition from reptiles to aves. Okay, so this is the another morphological evidence. Let's talk about the next evidence, which is your embryological evidence. So far we have completed one evidence, anatomical or morphological. Second is embryological. In embryological evidence, there was a scientist named Von Baer. Von Baer did embryological studies. Embryological studies on vertebrates. So this chart that you see, this is a study of a scientist known Von Baer. So Von Baer saw the various types of stages of like here different types of organisms. Some are amphibians, some are fishes, and some are reptiles, and some are mammals. Okay, so if we'll clearly see, so imagine this is a fish. Okay, these are amphibians. Second one is amphibian. I'm writing the short form. Okay, this is fish, amphibian. This one is a reptile. This one is a bird, and these one are mammals. Okay, so you will see that if we'll compare the embryo, if we'll compare the embryological stages, this is first stage, this is second stage, this is third stage. So you will see the embryological stage of fish and even mammal, they matches. The initial stages, early embryonic stages of all the organism, they matches. Now let's see the next one. This stage of a fish and this stage of mammal, does this match? No. But yes, this matches with this one. Alright. So Von Baer just does this study and didn't tell anything about evolution. This study was read by some other scientists. The name of the scientist was Ernst Haeckel. And Ernst Haeckel gave biogenetic law. Ernst Haeckel read the study of Von Baer and gave biogenetic law, which says ontogeny recapitulates phylogeny. What does it mean? It says that ontogeny means embryological studies. What does it mean? Embryological studies repeats, recapitulate means repeats phylogeny. Phylogeny means ancestral history. So when Ernst Haeckel read this study, he came to the conclusion that the embryological study repeats the study of ancestors. How? The first evidence he gave that the embryo, early embryo of all the organisms, they are same. Okay, this was what he said. Let's write what Ernst Haeckel said. First thing he said, all early embryos are same. All early embryos are same. That means the embryo of fish, amphibian, reptiles, birds and mammals, they all are same. Okay. Second thing, he said that all embryos have notocode. If it is the embryo of fish, if it is the embryo of bird, it's embryo of mammal. Every chordate have notocode, at least in one cycle in their life or one part in their life. This is what we say about chordates. Why are we chordates? Because we also have notochord, but when we are in the womb. So notochord is one character. So notochord is present in all the early embryos. In some, in the initial stage it gets replaced, in some in the later stage it gets replaced. Okay, this was the second. Third example, he say that mammals pharynx have gill slits in embryonic stages. What does it mean? When we were embryo, our pharynx, it was formed by the fusion of these pharyngeal gill slits, which are functional in the fishes. So technically, he sum up that. By this, what does it mean? If it is saying mammals have gill slits, which are functional in adult fish, this is what it say. So he tried to say that one organism's adult's character is seen in the embryo of another organism. Like the adult fish character is having gills, is seen in the embryo of mammals, which was completely rejected by von Baer. Von Baer said this, nothing happens. One organism's adult characters cannot be seen in embryo of the another organism. So by that, von Baer rejected this. So let's read this line of NCERT. Embryological support for evolution was also proposed by Ernst Haeckel based upon the observation of certain features during embryonic stage common to all the vertebrate that are absent in the adult. So embryological evidence, it was given by Ernst Haeckel. So how did it say? He explained this by telling about the embryological study of the vertebrates. Okay. And telling these common characters which are not present in the adult. For example, notochord is present in the mammalian embryo, but it is not present in the adult mammals. Okay. For example, the embryo of all vertebrate include human develop a row of vestigial gill slit just behind the head, but it is a functional organ only in fish and not found in any other adult vertebrate. So just I have told you, like mammals have gill slits in their pharynx. Behind the head, it's talking about pharynx, but it's functional in the fish. However, this proposal was disapproved on careful study by Carl Ernst von Baer. This is a full name of von Baer, Carl Ernst von Baer. He noted that embryos never pass through the adult stages of other mammals, right? So von Baer completely rejected this theory that no character is passed, no adult character is passed through the embryonic stages of the other organism. Okay, so that's about the embryological evidence which was given by Ernst Haeckel, rejected by Ernst von Baer. So we'll study the other evidences in the next class. Till then, bye bye, take care. Read the NCERT, although we have read a lot of it, but do read the NCERT of evolution, and I'll suggest all of you to read the NCERT after you complete the chapter with me, because there are a lot of things which are scattered in the evolution NCERT. So I'll suggest you read NCERT. You can read it if you want to read it now, but it will be better if you'll read it after we complete the chapter. So tata, bye bye, take care, work hard.